Welcome to another session of Putheti videos. The class begins. The play of adoptive immune system. We had seen in immune system that there are two kinds of immunities. One is innate immunity and one is adoptive immunity. When a virus or bacteria or a pathogen attacks our body, it is the macrophages and neutrophils of the innate immune system that face them first. It takes 1 to 2 weeks for the adoptive immune system to get activated. Till then, the innate immune system is in action. Now, it may be too confusing when T cells come to play, specifically CD8 and CD4 T cells. And among CD4 T cells, there are CD4 helper 1 cells and helper 2 cells, which are TH1 and TH2 cells. And when do B cells come into play? Do they act independently or together or serially? Let's take a look. For the adoptive immune system to get activated, there are macrophages from the innate immune system that also play a role. How does that happen? Now let's look into this situation where we have a macrophage and it is infected by a virus. And this macrophage is not able to defend itself. It's not able to kill the virus by itself. Now, what does it do? What it does is that while this virus infects the macrophages, gets its nucleic acids into the macrophages, uses the macrophage machinery to make its own progeny, and it is multiplying inside. We don't know anything that's happening from outside. So what happens is that the macrophage, although it is not able to defend itself, what it is doing is that it is picking up particles of the virus, the viral protein, and it is placing it in its own vesicle and it is transporting out to the surface. Why does it do that? Now here we have a macrophage presents viral particles which are very small in size. If you're looking at a peptide, maybe 9 amino acids long, they present it through MHC class 1 to the CD8 cells. So it is picking up small particles and it is presenting outside. Now, excuse me if these two fingers are very creepy. That's a cartoon that I made. I thought that would be helpful in memorizing better where a macrophage is actually presenting it. But we'll see how it goes. So once it presents this peptide to the CD8 cells, now CD8 cells, the T cells, they get activated. These are cytotoxic cells. What their role is to destroy the cell that is infected and prevent further proliferation of that pathogen, specifically this virus. Now, what it does is that it activates the nucleases production in the macrophage. Now, what do these nucleases do? They cleave the DNA and the RNA of the macrophage, the cell itself, and also those that are involved in the viral particle proliferation. So now these nucleases destroy the DNA and RNA and whatsoever that is there, and that prevents the viral proliferation. And the CD8 T cells also make granzyme B and perforin, which go and puncture the cell and destroy the macrophage. Basically, it kills the macrophage. So this is how the CD8 T cells get involved in the adoptive immune system. Now let's look into what the CD4 T cells do. In case of CD4 T cells, you have two kinds of cells. As I said, there are TH1 cells, T helper 1 cells, and T helper 2 cells. Now let's focus on T helper 1 cells. Now, when a macrophage takes up a bacteria and breaks it down, and it's actually not able to kill it completely. So that's where the adaptive immune system is coming into play, because innate immunity is not sufficient to clear this pathogen. Now you have a bacteria that is well engulfed, but this macrophage is not able to destroy it because its lysosome is here, but it's not merging with its vesicle where you have a pathogen. So what the macrophage is doing, just in case of CD8 T cells, is that it is presenting bacterial peptides onto the surface. Here you have MHC class 2 that is presenting to the CD4 TH1 cell. Now what this CD4 TH1 cell is, it is antigen specific. Just in case of CD8 T cells, these are antigen specific. If it was a different antigen, CD8 wouldn't have acted, because this CD8 is specific to this peptide. Now you have a T cell receptor on the CD4 cell that is very specific, that is absolutely specific to this peptide. Now that's when an activation of CD4 TH1 cell happens. It got signal 1 through MHC and signal 2 like CD28 or other molecules. We will look into those details in later videos. Now you have CD4 TH1 cell, and then what it does in return is that it gives a signal that would help merging the vesicle with the lysosome, and that will result in the breakdown of this bacteria. So CD4 TH1 cell is actually helping the macrophage in destroying the bacteria or the pathogen that is there inside. This also gives signals that would activate antibacterial agents in the macrophage. That's how CD4 TH1 cells work. Now let's look into CD4 TH2 cells. Now this is a lengthy process, because just in case of here, you have a CD4 TH2 cell that is very specific, that is absolutely specific to this peptide. It gets signal 1. It also gets signal 2 from here, and this gets activated. Now this CD4 TH2 cell is activated, that is activated specifically to this peptide, this antigen. Now this goes and activates a B cell that has already identified this antigen in the circulation or in the secondary lymphoid organs. Now remember, CD8 T cell action can happen in the blood or in secondary lymphoid organs, but CD4 and B cell activity takes place in secondary lymphoid organs. Now this B cell also received this peptide from the macrophage because it's all in the secondary lymphoid organ. So now this is specific to this. This B cell has an antibody on its surface that is specific to this peptide. That's how it can bind. Now it has bound, but that is only signal 1. It needs a signal 2. And that signal 2 is provided by CD4 TH2 cell, because now this is activated. Now this will give signal only for that peptide, that antigen. So it gives a second signal to B cell. Now what does that happen? That causes an activation of B cells. And now this B cell will become a plasma cell, and once it becomes a plasma cell, it starts making antibodies. What kind of antibodies? Specifically this antibody. That is the only antibody that this B cell can make. That is the only clone of antibodies this B cell can make. Now these antibodies, what they do is, once these antibodies are produced in huge amounts, they go and attack this bacteria that is there in the circulation. And now once these antibodies go and bind to this bacteria, that will activate complement cascade, that will activate the complement system, and the complement system will come and puncture this bacteria and destroy this bacteria. This is how a CD4 TH2 and B cells get involved in the clearance of a pathogen from our immune system. And this whole art was done by me, so that's why you see my signature here. I hope this play of adoptive immune system was interesting and it is clear to you. If you have any questions, please send it to me in the comment section. Please subscribe to my channel and please like my videos and give your feedback. Thank you very much.